Hello. So today we are going to look into the topic of regulation of gene, which is a very important aspect of. We have studied protein synthesis, and so we know now that the genome, the DNA, contains the structural protein also and the functional protein also. So if there is a structural protein and the functional protein, then there is a structural gene and a functional gene as well. Now, each cell in a multicellular organism will have the same DNA. Then, you wonder that if they have the same DNA, then why are they functioning differently? And why is that cells in different parts of the organism's body exhibit different characters? So, if there are this is one cell and this is the other cell in the body, in the human body. So, they are in the same human body, so they have the same DNA, but they separate. They are different, they are distinct, they function differently. So if one of them is the stem cell, the other could be the muscle cell, and the functions are different. So why is it so? It's not only in the eukaryotes but also in the prokaryotes, like bacterial cells. Like bacterial cells from within two pure cultures. Like, what is a pure culture? Like, you know, it's a system where I'm growing the bacteria, and all the cells are of the same bacteria. There's no other cell in this. So this becomes a pure culture. So if I have, say, two pure cultures and I choose any of the bacteria, or we are talking about all of the bacteria from the same cell. So if this is E. coli and I grow them separately, okay, and they are pure, they don't have any other contamination, all the cells are of E. coli. But if I expose them to different environmental conditions, for example, I expose this to extreme heat and I expose this to extreme pH, so alkaline pH, will they behave the same? No, they will not, because the environmental condition will dominate the expression of protein. So the expression of proteins is dependent on the environmental condition to which it is subjected. So here some proteins might express, some proteins might suppress in both the different conditions. So regulation of gene, understanding this whole phenomenon is very important, because even if the cells are genetically identical, there are certain proteins which are expressed, which are not expressed by the same set of genes. Only a subset of proteins, this is the right word, only a subset of protein is expressed at a given time. This is because the environmental conditions, the physiological need, we have already discussed that, they all dominate the regulation of the gene, or they all determine the regulation of gene. Okay, so in short, like, what is gene expression? It is making of a protein. We know it's a polypeptide. We have studied it in protein synthesis. So making of a protein by using a gene, which is on the DNA. So when we're talking about gene expression, the gene expression in total, it contains about transcription of DNA, trans- translation of mRNA, and then post-translational modification. There is also post-transcriptional modification, and in eukaryotes and in prokaryotes, it's different. In eukaryotes, the gene regulation is a bit complicated. In the prokaryotes, it is mostly observed in the transcriptional stage. In the eukaryotes, also, transcriptional stage and the post transcriptional um, modification. These are the two places where the gene expression can be regulated and is regulated, because once the mRNA is formed, the protein is bound to be formed. So before the mRNA is formed, the transcription is the stage where the regulation can happen there. Okay. Now, yes, we're talking about regulation during transcription. So there are these two concepts, regulatory proteins. These are also called as transcription factors. So transcription is controlled by regulatory proteins. These are very important. Okay. Now, these regulatory proteins, they can bind to the region of the DNA, and this region of the DNA is called as the regulatory element or regulatory elements. In a DNA, there can be many such elements or a single element also. And where are these regulatory elements located? They are located always near the promoter. Okay, so we know, we have already studied that where is the promoter. We are also going to see that in the next slide. Now, what is a promoter? Something that promotes, something that initiates. The name itself tells you that. So it's a region on the DNA where RNA polymerase binds to initiate transcription. So what happens if there is no promoter in the gene? If there's no promoter in the gene, the whole expression of proteins will always be on a switch on mode. Okay, it'll always be on a switch on mode, and the cell will always keep on producing the protein even if it is not necessary for the cell. Okay, now in the prokaryotes, the energy conservation part, like the cell does not want to waste its energy on production of proteins which is not required by the cell. It is only produced when the cell requires it. So if there's no promoter, the production switch is always on and the cell will get exhausted and it might just wear off. So promoter is a region of the DNA where RNA polymerase binds to initiate transcription of DNA to mRNA. So it's like a switch. The regulatory protein that is formed will go and bind on the promoter region. The switch is on. Okay, only after regulatory proteins bind to the regulatory elements, proteins can interact with the RNA polymerase, and hence then the whole process starts. So when we talk about regulatory proteins, there is activate. They can be of two types. One is the activator and one is the repressor. Now, what is activator? Activator is a protein which will go and bind to the promoter, and then the promote it'll initiate transcription. Repressor is preventing, it suppresses the transcription. Now, are there only regulatory proteins that take charge of this whole gene expression thing? No, there are some things which are also called as enhancers. Enhancers are additional, okay, but they are still very significant. These are some factors apart from the regulatory proteins that are produced in the cell. Apart from that, enhancers are very, very important. They also play an important role in the process of the regulation. Now, the genomic DNA contains of the regulator gene and the structural gene. See, we saw that there are two types of proteins, regulatory proteins and structural proteins. So if these proteins are produced, it means that the genes also have to be there. So if you have, say, this DNA here, and you will see a part of it is regulatory gene, a part of it is structural gene. So what happens is, now this is the promoter, this is the terminator, and then in between you have the sequence, the DNA sequence that needs to be transcribed, then it will be translated, and then the protein will be formed. So this is a regulator gene. Now this gets translated to an mRNA, and this mRNA codes for this regulatory protein. Now this regulatory protein can be of any structure, and then this will go and bind on the promoter. Okay, this will go and bind on the promoter of what? Of the structural gene, and then it will certainly cause whatever it is expression of the other gene. Okay, and then this is a structural gene here. This part, this is again a promoter, this is a terminator. The part in between the promoter and the terminator is the area of our interest, and then it gets translated to mRNA, and a functional gene is formed. So a functional enzyme is formed. Okay, so regulator protein and a functional protein or a functional enzyme. All enzymes are proteins except one, that is ribozyme, we know. So these are the two important genes in the genomic DNA which are responsible for the production of regulator protein and the functional enzyme. Now we saw that what are regulatory proteins? They can be activators or they can be repressors. Okay, if they go and bind on the other gene, activators go and bind on the other gene, they can promote transcription. Repressors repress or prevent transcription. And as a production of the functional enzyme is the next step actually. So what are the factors now which control or regulate the gene expression? These are more or less environmental factors, mostly. We saw that like there are two pure cultures of E. coli in the beginning of this presentation. I explained that if you expose one set of pure culture, say to a high pH, and one set of pure culture to high heat, then will they behave same? Will they be able to produce the same set of proteins? No, because the environment are different. So at high pH, some of the genes might express, some of the other genes might repress, whereas in a high temperature something else happens, some other proteins are produced. So environmental factors determine the protein, environmental factors regulate the gene expression. Okay, now when we are talking about gene expression, there are two types. One is positive and one is negative. We are going to see that in the next slide now. So positive gene regulation, negative gene regulation makes, so when we are talking about positive gene regulation, these genes will synthesize proteins which will help in gene expression. Okay, so how? Now, this is a segment. You have a promoter gene here, you have an operator, you have this area that needs to be transcribed. This is the sequence that will be transcribed, translated, and you will get your functional enzyme here. Okay, so if the positive regulators are formed, so what will happen is, if the positive regulators are formed, that is, what is a positive regulator? It can be termed as an activator. So what happens is, the activator will go and bind on the operator. Okay, and if the activator is a positive protein, then it activates the system, it activates the gene regulation, and transcription will happen. Now if the activator does not bind to the operator, okay, if the activator does not bind to the operator, there will be no transcription. All right, so there will be no formation of your protein there. So positive regulators are responsible for the activators, because they themselves are activators. So positive regulators are also alternatively called as activators. They usually bind in the promoter region, okay, and will allow the efficient transcription when on association with RNA polymerase. So it's not only the activator that goes, that is doing the transcription process. Activator will have to come in contact with the RNA pol, then only the efficient transcription will happen. Okay, when we talk about negative gene regulation, it is a process which represses the gene expression. So it means that there is a protein which is called as a repressor, and that repressor will go and bind in the operator region. When this repressor binds to the operator region, there will be no transcription. But when this repressor does not bind to the operator region, then there will be transcription. So negative regulators, which are also called as repressors, will bind to the promoters and block. Okay, they block whom? They block the association of RNA pol, or they block the access of RNA pol. So if the repressor is bound to the operator, then RNA pol cannot bind here. So if RNA polymerase does not bind here, then the transcription process does not happen. So in this case, in case A, repressor has bound to operator, so no access to RNA polymerase. So no RNA pol, no transcription. When the operator is not occupied and the repressor is formed, but they cannot come and bind to the operator, then there is access to RNA pol. And RNA pol will come and bind here on the operator. So this is the RNA pol which will come and bind to the operator. And now the signal is received that transcription can happen, and then the initiation of transcription happens. Okay, so there are two types of genes in the genomic DNA. What are they? Regulator gene and the structural gene. Regulator gene is responsible for the synthesis of regular protein. Regular protein are two types, activators and repressors. Activators promote transcription. Repressors prevent transcription. The structural gene is responsible for the formation of the functional enzyme, and this functional enzyme is eventually the in the best interest of the cell. Environmental conditions predominate the expression, regulate the gene expression. There are two types of control. One is positive control, one is negative control. So positive control is production of activators. Activators will go and bind to the operator, and then the RNA pol is allowed or has access. So RNA pol will come and bind on this complex, and then the transcription will happen. If activator does not come and bind to the operator, RNA polymerase cannot come. It has no access. Okay, and then no transcription. Similarly, in negative, the protein that is produced is called as a repressor, and this repressor will repress the gene expression. It will not allow the gene expression, because once the repressor and operator complex is formed, there will be no access to RNA polymerase. No RNA polymerase, no transcription. Okay, but when the repressor protein does not bond or bind with the operator, then it is then RNA polymerase has the access, and then the transcription will process. Okay, so this is basically a little bit about gene expression. Now, why is it so important to study gene expression? Whether it is in the eukaryotes or whether it is in the prokaryotes, it still has a lot of significance, and there is still research going on it. So, as I mentioned, that gene expression in the multicellular organisms is more complicated, okay? Whereas in the prokaryotes it is less complicated. And if in the multicellular organisms, um, gene expression is responsible for probably cellular differentiation. Okay, so because stem cells they differentiate into different types of cells, so that you can call it as cellular differentiation. Now when we're talking about prokaryotes, so I'll write here as multicellular, in multicellular cells or in organisms, sorry. In a single celled organism like prokaryote, the main aim of gene regulation is to stop wastage of resources. Okay, in the next part we are going to see lacoperon. Lacoperon is very useful if this is a cell and if this is present in an environment which contains lactose. Lactose is a disaccharide. Now this cell will require proteins to utilize lactose and break it. Okay, break it into glucose and galactose. Glucose is the one which will be utilized by the cell. But suppose if glucose is absent and lactose is provided to the cell, in such a condition, what will the cell do? Will the cell die because there's no carbon source? No, the cell has a set of genes which we call it as operon. It is known as lactose operon because it is induced or it is activated by the presence of lactose, and only then, only when the surrounding environment has lactose, those genes, those set of genes express. Okay, when there is absence of lactose, those set of genes don't express. So the cell is very clear about its need, and in absence of certain inducers it will not produce that protein. So stop wastage of resources, okay, and control protein synthesis is the main aim. Now what happens because of uncontrolled gene expression? What will happen? Uncontrolled gene expression in eukaryotes will cause cancer and other diseases. There is certain, there is a malfunction basically. Okay, it's a stepwise process. Any malfunction in any of the step causes uncontrolled cell growth, and it might lead to cancer and other diseases. Now knowing the gene expression control mechanism is very significant, and that is what scientists are still understanding, and a lot of research is going, because it plays a crucial role in understanding human health, okay, and also we are talking about, say, human and pathogen interaction. There are a lot of diseases that are transmitted or that happen only because a certain pathogen comes in contact with a human. A pathogen is a disease causing agent. So this pathogen and human, they interact, and that's how the disease happens. So if we understand the gene regulation process, how to control the expression of a certain protein, that will be very significant in understanding the diseases that happen in humans, okay, the genetic diseases that happen in human, and as well as the diseases that are happening because of the human and pathogen interaction, okay. When gene regulation we are talking about in eukaryotes, again, we say it is complicated. It is complicated. So what happens is basically it is all working on signals. So the cell in a certain environment, the cell interacts with the external environment, okay, wherever, in whichever condition it is. Say it is at high temperature, high pressure, high high pH, or any high sugar, okay. In all those conditions there is an interaction happening between the cell and the external environment, and because of that, then the cell starts sending some signals. There are certain molecules that are produced in the cell as an outcome of this interaction. Then the DNA also gets the signal, okay. So this interaction will lead, because of the signals, uh, there is an interaction that will lead to expression or suppression, okay. Expression of certain genes, suppression of certain genes, this is all determined by circumstances, in short, the environment. So the environmental conditions play a very significant role in the gene expression process, okay. Okay, thank you. I hope you have understood. If you have any questions, please leave your questions in the comment section. Thank you.